In 2021, 500,000 children were born with sickle cell disease. Those children will grow up to have a life expectancy of 52 years for males and 49 years for females. This fatal disease has taken away lives from 376,000 people in a single year. Sickle cell disease is caused by a mutation in the protein hemoglobin, which is responsible for carrying oxygen into our blood. The mutated bad gene causes the red blood cells in our body to be malformed, unlike the soft and round blood cells found in healthy people. Thus, these weirdly shaped blood cells are called sickle cells. These sickle-shaped cells can't move easily through the blood vessel, meaning that they can clump together and block blood flow, causing pain and damage to organs. Now, as technology advances, scientists have discovered gene editing tools that have the ability to edit the genetic coding in human cells, thus being able to cure genetic diseases such as sickle cell disease. An example of a genetic editing technology is Casgevy, an FDA-approved CRISPR-Cas9 therapy that works by extracting stem cells from a patient's bone marrow or blood. These extracted stem cells are put into a laboratory, and Cas9 is used to edit the genetic code, suppressing the production of the bad gene. The edited cells without the bad gene are then infused back into the patient's bloodstream to begin producing the healthy genes. Emmanuel Charpentier and Jennifer Doudna won the 2020 Nobel Prize in Chemistry for developing the CRISPR-Cas9 technology. This new form of medical treatment is already shaping how we treat diseases and will impact the future of medicine. Hence, it is crucial that we understand their advantages and their limitations so we can make informed decisions on how the technology should, and more importantly, should not be used. For those who are unfamiliar with this topic, Cas9 is also referred to as genetic scissors, a genome editing device that precisely cuts DNA and then renders natural DNA repair to modify the gene in our desired manner. So, what does this mean in practical terms? This tool enables scientists to make targeted genetic changes, offering potential treatments for genetic diseases, such as sickle cell disease, previously mentioned, but also offering the possibility of developing cancer therapies. However, as many say, with great power comes great responsibility. This brings us to many ethical concerns surrounding CRISPR, where the pursuit of scientific advancement clashes with its potential consequences, particularly the ethical concerns regarding germline editing, where the DNA in reproductive cells, such as the sperm, egg, or embryo, is changed. This means that any edits would be inherited and passed down to future generations. The pros are that theoretically, germline editing is able to eliminate hereditary diseases before birth. This means that, let's say, a family with a history of diabetes could have their disease permanently edited out, preventing their children from inheriting it. On the other hand, this also means that the new genes, after being edited, have the power to reshape the genetic composition of entire populations over time. As normal genes change and adapt to your bodily needs, edited genes can exist steadily and can be inherited unless due to mutation or other factors. In contrast to germline editing is somatic editing, which involves the alteration of DNA in non-reproductive cells, meaning that the changes are limited to the individual being treated and are not passed down onto future generations. Casgevy, the treatment for sickle cell disease, is an example of somatic editing using Cas9, where the edited genes would only affect the person undergoing the treatment. As of 2024, CRISPR is legal in the US. Many hospitals and biotech companies are conducting clinical trials with the regulation of the FDA, and if successful, treatments would be available for the market. Most clinical trials that the FDA regulates are of somatic editing, as federal money cannot be spent on germline editing due to the many ethical concerns. With that said, scientists in other countries are abiding by different regulations. I believe that many here have heard of designer babies, where CRISPR is used after fertilization to edit the genes in the embryo. many people are finding these DIY babies intriguing, as you can handpick your baby's sex, IQ, and even genetically modify it to make it free of diseases. Sounds amazing, right? Now, the fear isn't just about how the technology works, but rather about being accountable for the far-reaching consequences of the technology. In 2018, Chinese scientist He Jiankui created the first gene-edited twin babies that would be HIV resistant. Rather than being showered with praise for this amazing-sounding technological advancement, He Jiankui was sentenced to 3 years in prison and faced an immense amount of criticism from scientists and ethicists around the world. This is because of the many ethical concerns that arise, such as the potential for the edited gene to make the twins more susceptible to other diseases, such as the West Nile virus, the edits being pointless as not all cells may have been able to carry out the intended edits, and also concerns over unintended edits in other parts of the genome that may lead to cancer. Ultimately, the biggest concern over CRISPR would be germline editing, the fear that the mutation in the twin babies would be passed on to future generations. Germline editing and mutation are scary because it has the potential to introduce long-lasting changes in the human genome. In other words, it is possible to create an entirely new human species. Acknowledging the need for gene editing technology, as many parents may not want their unhealthy genes passed down to their children, but also realizing the limited knowledge scientists have over the consequences of permanent gene alteration, many other technologies and methods are available to resolve this issue. An option family with genetic diseases can pursue is in vitro fertilization, also known as IVF. In IVF, a patient is stimulated to produce eggs using a fertility drug. The eggs are then retrieved and fertilized with a sperm in a laboratory. For parents with genetic disorders, IVF is done after pre-implantation genetic testing, PGT, which allows doctors to screen embryos for specific genetic mutations before implantation, ensuring that only the embryos free from the disorder are used in the process. IVF allows parents to have biologically related children without having them pass on inherited diseases. This technology has been successful for 4 decades and have accounted for 2% of all births in the US in 2018. Another alternative for families with genetic diseases is to get an egg or sperm donation. In this process, an egg or sperm can be chosen from a large bank and can be selected based on characteristics such as IQ, appearance, education, and health history. The donated sperm or egg would then be fertilized using insemination or IVF. This technology allows parents to have healthy children and also opens up a path to parenthood for same-sex couples or single parents to have children. As we stand at the frontier of revolutionary medical technology, it is up to us, scientists, policymakers, and members of society to ensure that these tools are used responsibly. With DNA being discovered merely 50 years ago, we must proceed carefully. For scientists to strike a balance between innovation and caution when finding cures for diseases such as sickle cell disease or HIV without setting the groundwork for disaster. And for non-scientists, to temper our expectations for exciting new technology, as while many may hold the potential for amazing advancements, we must ensure that the technology serves to heal rather than harm. The choices we make today will not only determine the course of medicine, but may ultimately define the future of human genetics. Thank you.